I thought it would be helpful for me to pull out some of the things that are really important in this packet to make sure that we're really understanding what translation is. The replication and transcription packets, I think, is a lot simpler because we're really just swapping one letter for another. And remembering with DNA and RNA we have the T and the U. But beyond that, it's not nearly as complicated as translation. So I just wanted to go through a little bit from this packet and really what I want you to take away from this, most importantly. Now, we did talk about transcription in the last packet, which is going from DNA to RNA. This is what you would generate from the template strand of DNA. You would generate this mRNA molecule, which would leave the nucleus if it's a eukaryotic cell and move to a ribosome. The things that you need to really pay attention to here, we have a sequence which has A, U, G, and C. Those are the 4 letters in RNA. We have this methyl G cap, and we have this poly A tail. Now, it does explain more of that in the text, but I really want you to just be able to recognize what mRNA does and what mRNA is and what mRNA does. So this is the code in order to build a protein. The methyl G cap is what the ribosome uses to say this is something that I should be translating. The poly A tail sends a signal to these ribonucleases that the mRNA, the mRNA should not get broken down. The job of ribonucleases is to break down RNA. Lots of times that RNA is viruses that may invade our cells. So we've evolved these enzymes, ribonucleases, to break down RNA that shouldn't be within the cytoplasm. Coronavirus is an RNA virus. Cold virus is an RNA virus. HIV is an RNA virus. It does make sense for ribonucleases to be in our cells. This poly A tail does not code for anything. What this does is tells the ribonucleases, don't break me down, I'm not viral DNA. This is the ribosome. I wanted to make sure you understood. There's 3 parts, 3 sites within the ribosome, there's a large subunit, a small subunit. They're made of RNA and protein. A site, P site, E site. This I won't actually test you on. I just wanted you to be aware of this. What you need to know is that there are 3 stages to translation. First is the initiation phase. That's where your mRNA comes in to the ribosome, and this start codon says this is where you start translating. There may be other letters actually ahead of this start codon. This makes it a little bit simpler, but this is the start codon that says now you need to start bringing in amino acids. The tRNA has to go out, get the amino acid, bring it to this spot so we can start making a polypeptide by linking these amino acids together. This is initiation. Initiation is starting. Elongation. Now again, you don't have to worry about what the P site, the A site. When you get to AP Bio, you might have to worry about that. Not for this class, but you need to know there's an initiation phase where everything gets started. An elongation phase where we're making a polypeptide, which will eventually become a protein. And then we will eventually have a termination phase. During the elongation phase, every group of 3 amino acids is going to be the code for a amino acid. Every 3 letters in the mRNA is going to be the code for 1 amino acid. That's how, with only 4 letters, we can actually code for 20 different amino acids. What happens is this is the code, this is the signal, this is the start signal. This is the next one after that. This will tell me that proline should come in. And then the tRNA, which is connected to proline, has the complementary code. We call this the codon. That codes for the amino acid. We call this the anticodon. That tells the tRNA that it's in the right spot. But these are the letters that actually tell us the code. The code is on the mRNA. Elongation, you see we're putting together amino acids. That's what I wanted to make sure you understood. The initiation phase, you bring in a methionine. You get the process started. You get the protein started. Then we start putting together amino acids. We're making the polypeptide longer. That's elongation. And termination is where the ribosome gets the signal. UAA, time to stop. That's when the protein is now ready to be exported from the ribosome, and this ribosomal subunit, the large ribosomal subunit, small cell ribosomal subunit will oftentimes come apart. The tRNAs have no more of a job to do. This mRNA could get broken down, or it could just go into another ribosome and start the process again. So we have initiation, elongation. Termination is when the protein has stopped being made. Now this is where it might get a little bit confusing. I want to make sure that we understand what we need. And in fact, I probably should cut this out, but I won't at the moment. What I do need to do is separate out my key. What I understand and what I should actually probably be transferring this to another sheet of paper. There was a whole lot more to cut out on this sheet, but I decided not to force you to do that because I think we've done enough cutting out with the other things already. Now this may not seem immediately logical. I will show you two different ways that you could go about this. We'll do the easy way, the very straightforward way first. This is the mRNA. This is the mRNA that goes into the ribosome. As we said, we have a start codon. Start codon is AUG. That tells the ribosome to start sending out tRNA, getting these amino acids, and bringing them back to make a protein. AUG is the first one. That's the start one. What this would code for, AUG, is methionine. That's the first amino acid. It would extend down here. That's the codon. Every codon is 3 letters. The next one, GCA, will be here, alanine. Covers the next 3 letters. Next 3 letters, AAC, that's abbreviated as ASN. So the next amino acid is right here. Then GUU. So this is there is actually a code here. The first letter is a G, second letter U. So GUU, third letter U. GUU. Now I get to UAA. UAA is a stop codon. It does not code for an amino acid. It tells the ribosome, we're done. You did your job. The ribosome comes apart, releases this mRNA, which may go on to meet another ribosome, or this mRNA could possibly be degraded. This is your poly A tail. This is your stop. Nothing gets coded here. This is your poly A tail. Nothing gets coded there. All the poly A tail signals is to those ribonucleases, don't break me down. I am an mRNA that's supposed to be here. If you wanted to do this slightly more complicated, I'll bring it down here just so you can get a better idea. This is certainly good enough. But if you're going on to AP Bio at some point, you might want to find that this actually has an anticodon, the tRNA, which kind of has this twisty shape. It's a lot more intricate than that, but this would be attached to a methionine. The codon is AUG. AUG is the start, AUG is for methionine. On the tRNA, the thing that would recognize it would be the UAC. These would bind to tell the mRNA and the ribosome that we match up. There should be a methionine there. This one, the code for the amino acid, alanine, would be the GCA. If you really wanted to do the tRNA, that would be CGU. Those are connected on the tRNA, and that is connected. tRNAs do have a shape to them. Now, same thing here. AAC, that's the codon for our third amino acid. Whoops. The anticodon on the tRNA would be UUG. And the fourth one would be CAA. Same idea. This would be CAA. Is the anticodon. The codon is GUU, which we've seen before is, this is the codon for valine. This is the anticodon, which would be on the tRNA. Just to get a better idea of how this whole process works. Then we get to the stop. The stop means don't bring any more tRNAs. This is your stop. And then this is the last part, is the poly A tail. Which again tells those ribonucleases, don't destroy me. That is what you need to know for this packet, that these mRNAs code in groups of 3 for amino acids. And these combinations, all of these different combinations are all used in some way. Some are used to start, some are used to stop, and then some are used for amino acids. And you might guess that serine, for example, is fairly common in a lot of amino acids, in a lot of proteins. That's why you have a lot of different codons that would code for it. Same thing with proline. So that is kind of the idea. Some of these are not nearly as common, therefore there aren't as many letters coding for it. But we do need 3 letters to code for each amino acid in order to get sufficient code to cover all 20 amino acids. That's what you need to know basically for this video, and I'm going to add one more about what happens if this code gets mixed up.